Aujourd'hui, nous avons le plaisir de recevoir Robert Hazen, sorry for the pronunciation, it's not good, de la Carnegie Institution de Washington DC. Donc, il va nous parler de Necessity, Chance and Origin of Life pendant 3/4 d'heure, et puis ensuite nous aurons une séance de questions. Donc, merci Robert.
Merci, thank you, Gail. Thank you all for being here, and it's a pleasure to be in Paris again and speaking about something that I find extremely fascinating. In fact, I think that all of us, because these are deeply philosophical questions, ask these questions about the cosmos. Are we alone? And is there a way that we can go into a laboratory and study life's origins, perhaps reproduce life's origins? This question of life existing on other worlds, perhaps near us, perhaps on Mars or other planets and moons within our own solar system. These are all questions that are being pursued rigorously. What I'd like to do today is look at three aspects of this. First, just introduce you to the basic idea of how we go about studying the origin of life in the laboratory, the scientific study. And there is a systematic way that people are approaching this. The second, I want to propose that this idea of chance versus necessity, which is so often brought up in the context of origin of life and other aspects of biology, that I believe this is a false dichotomy. That in fact we really need to be talking about continuums of probabilities, not just yes or no for a deterministic aspect. And finally, I want to demonstrate this rather new point, that at the scale of planets like Earth, events may be deterministic, which ironically could never or almost impossibly be produced within a laboratory time and spatial scale, simply because the scale and time available to an Earth-like planet is vast. And we'll see that quantitatively how that works. Okay, so let's begin with the scientific study. Some of this may be very familiar to you, but this is the way we think about it, and I do some of this origin of life research myself. First, basic assumptions. The chemistry of the origin of life, and the origin of life is a chemical event, has to be based on carbon. We're not going to have some exotic chemistry. The origin of life is a chemical process, and it uses the basic ingredients of a planet. You have air, you have oceans, you have rocks and minerals. And that's what you have to work with to begin with. And finally, that life's origin requires a sequence of chemical reactions. And it's the understanding of going from the simplicity, the relative simplicity of a geochemical world to the much greater complexity of a biochemical world that characterizes this study of the origin of life. So, let's think about chemical reactions, because that, as I say, is the basis. And when you think about chemical reactions, they always involve the rearrangement of atoms and their electrons. And there are many common chemical reactions, rusting, for example, cooking, using an antacid. And these are the basic reactions that occur in life. In the case of rusting, it's moving electrons through oxidation reduction reactions. In the case of cooking, we have polymerization and depolymerization reactions. We also have hydration and dehydration reactions. So here we're moving small molecules in those chemical reactions. And in the case of acid-base reactions, we're moving protons. So these are electrons, protons, and small molecules. That's exactly what life does. Very basic kind of simple reactions. We want to see how these reactions using carbon could have produced simple molecules in the early days. So think of this chemical reaction, a sequence of chemical reactions. First you have to make the simple building blocks, amino acids, sugars, bases, carbohydrates. These are the kinds of things. And then you have to organize, select, concentrate these molecules into biologically useful polymers or membranes. That's the second step. And finally, we have to have a cycle, a self-replicating cycle of chemical reactions. And for many origin of life researchers, that is the origin of life. As soon as you have that cycle of chemicals that makes copies of itself, then Darwinian natural selection can arise. So let me look at these in a little more detail. So the first step, just making those simple molecules. What you're trying to do is start with simple molecules, volcanic gases, for example, carbon dioxide, carbon monoxide, water, hydrogen disulfide, these simple molecules, and you want to make carbon-carbon bonds. that really is the key to the first steps in the origin of life. So going from CO2 to acetic acid to pyruvic acid, building up larger and larger structures. You can add nitrogen to this picture. You can add sulfur to this picture. And of course, this is the kind of thing that's been done now for 60 years and more. The original classic experiments by Harold Urey and Stanley Miller at the University of Chicago, using a simple tabletop glassware apparatus where you had water gently boiling. You had an atmosphere of various gases that were presumed at the time to be part of Earth's early atmosphere, and little electric sparks, which provided an impetus for more chemical reactions to form. And in the process of doing this, they were able to produce a whole variety of simple molecules, organic molecules, the building blocks of proteins, amino acids, sugars, making larger, more complex carbohydrates, molecules that played a fundamental role in metabolism, even the building blocks of nucleic acids. All these work, and the idea is this looked very much like a diet. This was so convincing to people, they thought this was the way the origin of life must have occurred. Of course, there are other ways you can do this. You can make chemical reactions in deep space, where you have what are called dense molecular clouds, like the Eagle Nebula, where there are very widely dispersed, but nevertheless a concentration of dust particles of various small molecules. There's also ultraviolet radiation, and that causes reactions. You can simulate these in the laboratory, as does Lual Mendola at NASA Ames Research Laboratory. So this is the kind of thing that's been done quite effectively, and showing, again, you can make amino acids, you can make sugars, you can make bases, you can make the basic building blocks of life. My own research laboratory, we look at black smoker environments, these volcanic vents on the ocean floor, where you have a very sharp contrast between the reduced conditions of the ocean floor rocks and the ocean itself. You have many particles, you have lots of surface areas, a very reactive environment, and again, you make all sorts of molecules. We use simple gold tube reactors. We subject these to temperature and pressure. We analyze them in various ways. And again, we find that we can quite easily make a whole variety of simple organic molecules. And recent studies even show that, quite surprisingly, that one can go into Earth's mantle, and under the correct conditions, temperature and pressure much, much higher than people would ever have thought when you think of organic chemistry. You can make a diagram here where you vary the redox conditions, the activity of oxygen and the activity of hydrogen, and you find that there are regions, even at these very extreme conditions of the Earth's upper mantle, where you can produce, under certain circumstances, hydrocarbons similar to what's in oil, or various carboxylic acids similar to what's in vinegar, depending on those conditions. So here in Earth's mantle, again, producing a rich variety of organic building blocks, the beginning steps of life. So this first step, chemical reactions of all different sorts in all kinds of energetic environments form the building blocks of life. That is a solved problem. It's something that we don't have to worry about too much. We can count on there being a rich inventory of molecules. So that's the first step. And in fact, it's almost too easy to make these small molecules. You make too many molecules. And here's what I mean. If you look at the inventory of all the different kinds of small organic molecules that are present, there are literally millions and millions of them. The current listing in a compilation called Beilstein is, I think, 7 or 8 million different small molecules. But if you look at a simple microbe like E. coli, it only has about 500 different small molecules. A very parsimonious selection, very limited. So the question is, you make too many kinds of molecules, how do you select, organize, concentrate just the right molecules out of that diverse mixture? So that's the second problem in chemical reactions. So we're going to look at those kinds of chemical reactions. Let me give you a sense of what I mean. In one of our very first sets of experiments performed almost 20 years ago, it's surprising, it's shocking to me that's true, we took pyruvic acid, that's a simple molecule with 3 carbon atoms, put it at high pressure with CO2 and water. And our hope was to make a 4-carbon molecule called oxaloacetate, which is a key step in many biomolecular reactions. So we ran this at very simple conditions. We thought we might just get a simple single product, but instead what happened is when you seal up the capsule with pyruvic acid, you run it for a couple of hours, and when you open those gold tubes, they're all puffed up. They have very high pressure inside. So you're a little worried you don't want to have an explosion, so you cool it down. You put the little gold tube in liquid nitrogen to cool it down. And it sits there until it's frozen, and then you snip it open, and even then it explodes. It just bang, and you know that there's been a lot of pressure or gas release. But it's still frozen. You put it in the bottom of a glass vial and let it sit there, and then after 30 or 40 seconds it starts to thaw, and you get this hissing sound, and this yellow, brown, oily goo starts pouring out of the capsule, and you realize that, and you made something very different from oxaloacetic acid. There's been a lot of chemistry going on here. And furthermore, then the aroma, the smell starts coming to you, and if it's a little bit lower temperature, it smells very sugary. It smells like molasses. If it's slightly higher temperature, it smells exactly like Jack Daniels. So, and it's amazing. I mean, you know you're making something interesting, and it just permeates the room. A room like this, you just have this overwhelming aroma after running just one of these little tiny gold tubes. So we make something very, very interesting. You do the chemical analysis, and what you find, this is what's called a gas chromatogram. So each of these peaks is a separate chemical. In fact, there are literally thousands of different molecules in this experiment. So we started with one simple molecule, and you make thousands. And for the origin of life, that's a problem. It's a bad thing. You don't want to make thousands of molecules. You want to make just a few very specialized things. So how in the world can this help us? So I contact my friend Dave Deemer and describe this to him. He's a University of California, Santa Cruz researcher, and he's a specialist in self-organization of molecules, self-assembly of molecules into these things called vesicles. And he said, have you tried putting that yellow-brown, oily goo into water? And I said, Dave, why would we want to do that? And he said, well, maybe they'll self-organize into small little features called vesicles. And indeed we did, and here they are. They're a few tens of microns across. They have an inside, they have an outside. These are just like part of the building blocks of cell membranes. So what we've done here is in spite of the fact that we made way too many molecules, some of them self-organize into a structure which is exactly what the origin of life required. You needed to have a separated compartment. And here's a way of making compartments in which chemistry can go on concentrated inside those little compartments. So it's certainly not the answer, but we find over and over again in experiments of this sort, some subset of the molecules makes vesicles. They organize. So that's sort of one example. However, there is a problem that many of the molecules that we're interested in are water soluble. They're not going to self-organize. They're not going to separate into membranes. So how do you select and concentrate those? Think about the prebiotic soup. It's much too dilute. The world's oceans are much too dilute. The chance of any two molecules coming together and interacting in the ocean is very small. So what you need to do is find a mechanism to select and concentrate molecules so that they can interact more readily. And one thing we've been doing is focusing on the surfaces of minerals. Now it's a little hard to see this, but these are tiny crystals of the mineral quartz, a very common mineral of beach sand. And if you magnify this and look at it closely, what you'll see is there's 6 little triangular faces that come to a point. You've all seen quartz crystals. But in this particular specimen, iron oxide has coated some of those faces, but only the alternate ones. And what this shows you is that the absorption of molecules onto surfaces is highly selective. Molecules may absorb to one surface that has a certain kind of structure, but they will not absorb to a second surface because it has a completely different structure. And we thought perhaps these mineral surfaces could be highly selective for organic molecules as well. And that's the experiment we've done. So we've looked at quartz faces where you see different structures at each face. We've looked at calcite, the similar sorts of things. These are common minerals at Earth's surface. Calcite's the mineral that forms clam shells and snail shells. It forms limestone reefs, coral reefs, and so forth. And what we found is you take a crystal like this, you dip it into a mixture of different molecules, Certain molecules absorb on one set of faces, certain molecules absorb on another set of faces. Indeed, you can separate left and right-handed molecules, molecules that are exactly the same except they're mirror images of each other. Now, this turns out to be a very important question in biology, because in our own bodies, in all living things, left-handed amino acids are selected over right-handed. Right-handed sugars are selected over left-handed. And so a question for a long time has been, how in the world do you start with random mixtures of molecules and yet end up with concentrated left and right-handed molecules? This is one way that you could do it using mineral surfaces. So we think we have some answers here. We can also understand in great detail using theoretical calculations why that's true. Here's, for example, part of the calcite surface, and an amino acid's coming in with 3 points of interaction on that surface. But if you take the opposite-handed amino acid, it doesn't fit very well, and you only get 2 points of interaction. Well, 2 points of interaction cannot select for a left hand or a right hand. Both the left hand and the right hand fit well. But if you have 3 points of interaction, one hand can fit, but the other one doesn't. So this is an explanation of why these calcite surfaces are so good at selecting left and right-handed molecules. It just has to do with the fit of molecules and their atoms onto the surface. Well, so this second step, I think we understand it fairly well, even though there are lots of details to be worked out. You can have molecular self-organization, and you can have selection on surfaces. So here's the way to select, concentrate, and organize molecules. I think minerals play a real fundamental role in this process. Now, the third step, and this is another kind of chemical reaction, is a sequence of chemical reactions that forms a cycle, a self-replicating cycle of molecules. And this is by far the most important and as yet unsolved problem in the origin of life. There are a number of theories, there are a number of ideas out there. One of them has to do with something that's very familiar to biologists. That's this idea of the Krebs cycle or the citric acid cycle. Sometimes it's called the tricarboxylic acid cycle, TCA cycle. And if you run it in the reverse way from the way we normally think of it, it's an engine of synthesis. You start with small molecules, acetic acid, CO2, pyruvic acid. You add small molecules one at a time. You go from 2 carbon atoms all the way around to citric acid, which has 6. And then the citric acid splits into 2 plus 4, and you have 2 cycles. Then you have 4 cycles. Then you have 8 cycles. And each time you go around this, you double. That's a self-replicating cycle of chemical reactions. And because it's at the core of so many cellular biochemical mechanisms, we think this may be a fundamental part of the story. I work very closely with Harold Morowitz, who's a theoretical biologist who's been proposing this for a number of years. So we've been working on this idea. But there are other ideas as well. Stuart Kaufman, who worked at the Santa Fe Institute in New Mexico, he proposed the idea of an autocatalytic network. This is a rather different idea. The idea is that the prebiotic soup had thousands and thousands of molecules. And some of those molecules would catalyze the synthesis of other molecules in the network. So every molecule in this network supports the production of some other molecule in the network. And so you're either part of that in crowd and you make more and more copies of yourself, or you're excluded and you gradually get winnowed away. You lose out to the network itself. So even though this is not exactly a cycle, this network has the properties of a self-replicating system. And then Kaufman's idea is you may start with 1,000 or 10,000 molecules, but gradually it becomes more and more efficient by winnowing away the outlier molecules and having a core, a smaller and smaller core, that may eventually fold down into something like the TCA cycle. The problem with Kaufman's idea is that it's virtually impossible to test in the laboratory. We have simply no way to analyze and monitor 1,000 molecules simultaneously, even if we knew what those 1,000 molecules are supposed to be. And we don't. Kaufman doesn't say that. He just calls them A and B and C and so forth. So that's not very helpful when you're trying to do scientific experiments. But it's still an idea that's out there. It's another kind of self-replicating system. And then, of course, you've heard of the RNA world. The RNA world where an RNA molecule, a piece of nucleic acid, has the properties that it can catalyze reactions, and at least conceivably, in principle, it could make copies of itself. So if you have a molecule that can do the catalytic work and carry information and copy it, then you have a single molecule that can act like a self-replicating system. So many people who come to origin of life from the perspective of a molecular biologist look at this. And I should give you a warning here, just as an aside. No one gets a PhD in origin of life. You get a PhD in chemistry, or mineralogy, or perhaps astrophysics. Some people get a PhD in molecular biology. And so everyone comes to the origin of life with a prejudice of what's important. So the molecular biologists all think RNA is the important thing. So they go to this. I'm a mineralogist, so I think minerals are important. Other people study lipids, or they study some kind of astrophysical, and that's where their focus lies. So the origin of life field is filled with people who come to it with kind of very narrow vision. I think we all have to expand our vision and recognize that the origin of life is not just physics or just chemistry or just geology. It's all of the above. And of course, it needs to draw on biology as well, even though the origin of life is not per se a biological topic, because it's before life. So there was no biology at that point. Okay, so these are different possibilities. There certainly are lots of them out there. We have not yet solved this problem. It's something that is the main focus of many laboratories around the world, and if there is a major news break sometime in the next 10 or 15 years, it will probably be the announcement that some laboratory has managed to produce a self-replicating cycle of molecules in the laboratory. And then if that set of molecules can mutate, of course, that's the real neat thing, because you have this self-replicating system of molecules. And if you have a series of chemical reactions based on organic molecules, it's very easy to have little mistakes creep in. And by that, I mean that instead of having a hydrogen in a certain position, you can have a methyl group. Instead of having an OH group, you can have a thiol group. Instead of having a hydrogen, you can put COOH, and so forth. You know, anybody who's done organic chemistry, you just shuffle things endlessly. And these different molecules will have very similar energies, and so it's quite easy to have mistakes. Once you have mistakes, then you have competition, and natural selection becomes inevitable, because in different environments, fresh water, salt water, warmer, colder climates, higher or lower pressure, different cycles will be preferred. They'll be more efficient. And so then you'll start having cycles competing with each other, cycles evolving. Competition is driven very, very rapidly because you select for the cycle that makes most efficient copies of itself. So that's our argument. That's the basic idea. So this first part, this is how we're approaching the origin of life in the laboratory, looking at these different steps and trying to winnow down the possibilities that an Earth-like planet can produce. And let me just point out one additional thing that's interesting here. We know on Earth there was one origin of life, at least. And it led to the biochemistry that every living thing on Earth shares. And that's really underscored by the fact there's only a single genetic code, and every organism uses the same code. But that does not preclude there having been many origins of life, perhaps thousands of origins of life on Earth itself, in which case the most efficient won out. Everything else became food. So this is something that's always in the back of our minds. Perhaps there was only one origin of life, but there may have been many. There may be many different chemical pathways. On a different planet, you may have had a completely different outcome. We simply don't know. That's one of the big questions. And that has to do with this idea of chance versus necessity, which I'm not going to come back to. I want to propose that chance versus necessity is, in fact, a false dichotomy. Now, you've probably read or heard of some of these ideas. So I'm going to be focusing again on chemical reactions now. So when I talk about origin of life, it's not just some sort of vague, fuzzy things. It's a sequence of chemical reactions. And I'm going to say that chemical reactions on Earth display a wide range of probabilities. So, how many have read Jacques Monod or know of him? Have you heard? So Monod wrote about these ideas in great sophistication. He's often kind of trivialized or perhaps pushed to aside by his last sentence: the universe is not pregnant with life, nor the biosphere with man. Man at last knows that he is alone in the unfeeling immensity of the universe, out of which he emerged only by chance. So that's kind of the encapsulation. It makes it sound like he's really saying life is a complete chance. It's an improbable, it's a frozen accident. Now, this was immediately countered by a number of people. Ernest Schopenhauer, anybody, you know this counter, anti-chance? Well, this is written 6 years later. He said the origin of life and evolution were necessary because of conditions on Earth. And so he basically is arguing that there's a necessity to life. So that's the antithesis. Is it frozen accident? Is it a cosmic imperative? What I want to argue is that those are extremes that don't reflect the reality of chemical reactions on Earth. I'm going to argue that chemical reactions on Earth display a wide range of probabilities. Now, it's difficult to determine what the probabilities are for organic molecules. But because of recent work I've done in mineralogy, it turns out that we can do very precise probabilities for the occurrence of minerals on Earth, and each mineral species on Earth represents a chemical reaction. So therefore, we can use minerals as a proxy for the probabilities of chemical reaction. Now, first, let me tell you what a mineral is: a naturally occurring crystalline compound that has a known crystal structure and a known composition. So every unique combination of crystal structure and composition represents a mineral species, and there are about 5,000 recognized mineral species. So we've been doing studies of mineral species that we call mineral ecology, a brand new field introduced last year in which we study the diversity and the distribution of mineral species. We can do this because we have very large databases. And so the question is, what are the relative roles of chance and necessity in Earth's minerals? So, what we realized was the distribution of minerals on Earth's surface is very much like the distribution of biomass in an ecosystem. So, take a redwood forest. And if you've ever had the privilege of walking through a redwood forest, you'll notice that almost all the biomass is in one species, large redwood trees. But the diversity is all these little things. And so if you want to understand the ecosystem, you have to understand both the biomass distribution, but also these small, rare species. And using those small, rare species, you can do all sorts of predictive things. You can, for example, measure the first plant you come to, and the second one, and the third, and the fourth. You measure 1,000, 10,000 plants. And as you do so, you'll discover new species at a slower and slower rate, something called an accumulation curve. So this is counting individuals. Here's the abundance, the number of different species that starts tapering off. You can also do this with genome data. For example, if you're studying the diversity of microbes. Again, as you count more and more individuals, you find an asymptotic relationship to the number. Now, this has never been done for minerals before, but we have now started doing this. It turns out the mathematics that we use, though, is not the same mathematics the ecologists use. It's the mathematics that are used by lexicologists studying the distribution of words in manuscripts. Now, you may think this is a pretty obscure or arcane way of approaching things. But because of terrorism, the National Security Agency in the United States has developed this mathematics in an incredible degree, so you can look at emails or other documents and assign authorship. And the way you do that is you see most words are very common, but rare words, the ones that occur only once or very few times in the document, define everything. They define the genre of work. They define the authorship. So you can tell, for example, if a manuscript was written by William Shakespeare or Flaubert or whoever you would like to, and this is a way that can be done quantitatively. So we've been taking the mathematics that have been developed in this way and applying them to minerals. And here's why we can do this. It turns out minerals also follow this, what's known as a large number of rare event distributions. There are a few minerals that are extremely common, that occur at tens of thousands or cover Earth's surface. In fact, 99% of Earth's crustal volume is fewer than 100 minerals. So those other thousands of minerals are very rare. They're very sparse. Some things occur at thousands of localities, some occur at hundreds of localities, but the probability is that a mineral will occur at very few localities. 8% of all minerals occur at only 3 localities. 22% of all known mineral species occur at exactly 1 locality, are unique to a single locality. And that means we can use this large number of rare event statistics. We have huge mineral databases that tell us the names of the species, their characteristics, and their localities. And that's what allows us to do this. So here's a distribution. This is a mathematical distribution where number of minerals occurring in exactly 1, 2, 3, 4, 5 localities and so forth. And you'll see most minerals occur at just a few localities, whereas fewer minerals occur at lots of localities. So we can use these statistics to come up with an accumulation curve. Here's the number of mineral counts. That means a mineral that's found at a locality. So calcite found at a particular quarry in the region of Paris, France. That's one locality data point. And we now have roughly 650,000 of these data points. New species appear here. And what you find is when you first start looking, you add new species very, very rapidly. Almost everything you find is a new species. But then the more you come, it starts asymptotically going off. That's where we were in February 2014 when we first did this analysis. And you can extrapolate into the future, and we can estimate the total number of mineral species on Earth. What it turns out is about 1,500 mineral species occur on Earth today, but have not yet been found. And we can actually predict what some of those minerals are. But for the purpose of this talk, here's the important part. You can also say what's the probability of each of those minerals occurring on an Earth-like planet. We have that distribution. And what we find is there's a number of minerals, almost 3,000 minerals, that are very, very likely to occur. More than 90% probability. Those are deterministic. There are also a number of minerals that have, maybe a 50% probability. They might occur, they might not. And there's some other minerals that have less than a 50% probability. And the way I interpret this, if you look at it, they're deterministic minerals, necessity. Any Earth-like planet is going to look very, very similar in terms of these minerals. There are extremely rare minerals, frozen accidents, things that occur only because of very weird combinations of circumstances. And those are unlikely to occur. So I'd call those frozen accidents or chance. But perhaps the majority of minerals just have probabilities. It's not their chance, it's not necessity, it's in between. Now remember, each of these minerals represents a chemical reaction. So on an Earth-like planet, you have chemical reactions that are extremely likely. You have chemical reactions that are extremely unlikely. But you have vast numbers that are somewhere in between, just probabilities. So it's a distribution, it's a probability distribution. Now we do not know whether this probability distribution for minerals is the exact same probability distribution for organic chemical reactions. But the hypothesis is that there will be some distribution with range, and that makes logical sense. So if the origin of life required 100 or 1,000 chemical reactions, the idea is some will be more likely, some will be less likely. That's the point I'm trying to make. Since each mineral species represents one of these chemical reactions, some are more likely than others. Exact same logic follows here. And so my conclusion is that it's not chance, it's not necessity. That's a false dichotomy. A wide range of chemical reactions. And that's something that's worth trying to understand in more detail, but it's something also that's a key to know. So let's go to the third part, and that's to demonstrate that events that are improbable in a laboratory. Some of these chemical reactions may be extremely difficult to reproduce in a laboratory, but given the time and the dimension of an Earth-like planet, it's easy. And what I'm going to do here is look at this hypothesis. Chemical reactions that are extremely improbable at laboratory scales can become deterministic at planetary scales. And to do this, I need to make a back-of-the-envelope calculation. So it's how many chemical reactions can occur on an Earth-like planet? How many kinds of experiments can we run? And here, it's a very simple calculation. you basically say, how long does it take for a reaction to occur in a mineral surface, and how much surface area do we have? So that's looking at the micro scale of an individual experiment. And then think about it, how much time do we have on an Earth-like planet, and how much surface area on which to run those chemical reactions. There are 4 numbers that we need to come up with. And I'm just going to run you through this quick back-of-the-envelope calculation. First, how long does it take for a reaction to occur? Well, that's something that's well understood. In many cases, it's a tiny fraction of a second, so you can have 1 million reactions on a surface in a second. In other cases, molecules will stick longer and therefore passivate the surface. But on average, I'm saying reaction times, even though they vary, 10 seconds per experiment is a very conservative number. So if you think about you having a mineral surface where a reaction can occur, every 10 seconds another combination of molecules is sampling that surface. So how much surface area does it take? That's the answer to question 1. Make a note of that. So surface area you have small molecules that are typically less than a few nanometers across. You have mineral surfaces where the repetition area is also less than a few nanometers. But let's argue that the surface area you need for a reaction is about 10 nanometers by 10 nanometers. That's actually a very large area for these small molecules, but again, a conservative number. So if you do that, that's the number, 10 to the minus 12 centimeters. I'm sorry that the bottom's cut off a little bit in this, but that's the second answer. So now we've answered the question about the time and spatial scale of individual chemical experiments in the prebiotic Earth. Now let's look at Earth-like planets. How much time do we need? Well, we know that there was life 3.5 billion years ago because we have clear fossils. Some evidence, even perhaps 4 billion years ago, life was present on Earth, and Earth is about 4.567 billion years old. So I'm going to sort of conservatively estimate that Earth had 600 million years. That's 2 times 10 to the 16th seconds. That's how much time we have to run chemical experiments. It may have been longer, and certainly on other planets you may have longer, but let's just use that number as a rough estimate. And finally, the fourth question is how much surface area? This is the most sort of involved, it involves the most assumptions, but you'll see that it's a vast, vast number. So we look at Earth. The total area of Earth is 5 times 10 to the 18th centimeters squared. Everything's going to be in terms of centimeters squared, so a little area like that. And how many of those surface areas are there? But remember, each square centimeter of Earth's surface represents a column of soil and rock. And soil and rock has a very large surface area because it has sand grains, it can have clay and tiny particles, volcanic ash, things like this have huge surface areas. The commonest near-surface mineral early in Earth's history and today are clay minerals. So if you look at clay minerals, it's typically 2 times 10 to the 6th, or 2 million square centimeters in each cubic centimeter. That's a mind-boggling number. Here's what it means. If you have a sugar cube size volume of clay minerals, it would cover a tennis court. So every cubic centimeter of clay mineral will cover 1 tennis court. Now, how much clay minerals are there? Let's just assume very conservatively that the average depth of soils on the early Earth was 1 meter. It could have been 100 meters in places, or 10 meters, but let's just say it's 1 meter. So that's 100 centimeters. So you have 100 cubic centimeters for every square centimeter of the surface of Earth, and that's a very typical kind of number, as I say. So you start to multiply this out. There's the surface of Earth times 100 cubic centimeters per centimeter squared gives you this immense volume, 10 to the 20th cubic centimeters of clay minerals, 500,000 cubic kilometers of clay minerals with surfaces on which you can do chemical reactions. And then you multiply that times a tennis court for each cubic centimeter. And it turns out we had 10 to the 27th square centimeters of mineral surface area. That's the answer to question 4. Okay, 4 numbers. All we have to do now is just do the simple calculation. There's the surface area of Earth in terms of reactive mineral surfaces. There's the time we have to play with. You have to divide that then by the surface area of a single experiment and the time for a single experiment. And when you run out that calculation, it's 2 times 10 to the 54th chemical experiments on an Earth-like planet. That's the number. 2 trillion, quadrillion, quadrillion, quadrillion experiments, just as a simple back-of-the-envelope calculation. Now that is more experiments than the average graduate student can do in 3 or 4 years. That's a joke. Okay, thanks. And so the conclusion is obvious. Many, many more chemical reactions can occur on an Earth-like planet. They can occur in a kind of laboratory experimental setting. So you can have things that are almost impossible to imagine ever having worked in a laboratory. It requires a very exact set of chemicals and surfaces and time and temperature, and all these things have to be juxtaposed just right. But that may be a key step in the origin of life. And it may never be reproduced in a laboratory, and yet it may have been reproduced millions or billions of times on early Earth, simply because there is so much combinatorial richness to it. So this is a conclusion that chemical reactions that are impossible or improbable at a short time scale of a laboratory may be inevitable at planetary scales. And some people might say, well, then why even bother? Even if life is a cosmic imperative, maybe we can never figure it out. But remember, we can use chemical and physical intuition to eliminate lots of possibilities and zero in on the ones that really will work. We can work backwards from biology because we know the biochemistry that works today, and so we can look at those specific chemical reactions. We can try new approaches. Combinatorial chemistry allows us to do, at times, millions of experiments at a single time, so one can perhaps approach it that way. And, of course, we need to hire more great students. I have lots of students I've been working with. They do amazing work. They have creative ideas, and this is happening all over the world in many, many laboratories. So, to me, this gives me great optimism that down the road we will be able to solve this problem. 3 conclusions. You can understand life's origin as a sequence of chemical reaction. That chance versus necessity is a false dichotomy. That Earth-like planets do things that we can't necessarily do in a laboratory. So what may be an improbable event at laboratory scales may be deterministic at the scales of a planet. So we haven't answered those big questions yet, but I think it's a great challenge. It's fun. It makes it great to get up in the laboratory. The things you don't know are what you want to drive you as a scientist. So we're going to still try. And with that, I want to thank my funding organizations, and thank you all for being here, and I welcome questions.
Thank you. Any questions?
I have a question of clarification. I know something about investigation of original life and based on the literature about protocells and the emphasis on compartments, important compartments. So can you go back and say a bit more on the relation between the appeal to minerals and the necessity to have those vesicles or proto compartments, and how they put the story about their integration to get some.
It's a great question. So, for example, Dave Deamer and Jack Shostak, who are working on these problems in the United States, Jack Shostak at Harvard, Nobel Prize winner for his discovery of telomeres, but he's actually been working on exactly this problem. When he just has compartments and the raw chemical materials, the carbon-based, he doesn't see much. When he incorporates clay mineral particles inside the vesicle, he starts seeing levels of organization that don't occur otherwise, including formation of RNA polymers and so forth. So the idea, once again, is, and I don't push minerals as being the only possible answer, but they do provide surfaces on which interesting chemistry can occur. So what many origin of life researchers, even people who are not mineralogists at all, are finding that they can accelerate, they can get much more interesting chemistry when there's these nanoparticles of minerals. It could be sulfides, it could be clay minerals, it could be other sorts of geologically relevant phases, which would certainly have been ubiquitous on early Earth.
So the idea would be that the surface of minerals would be a place where relevant reaction and materials could be encapsulated by independently self-assembling vesicles.
Yeah.
So the two events would converge on the surface of minerals.
Yeah. So what minerals can do many things for you. They can catalyze reactions. They can absorb and select, concentrate certain molecules out of solution. They can put those molecules juxtaposition so polymers can form. They can protect the molecules. What we found recently, for example, is that amino acids, which under these hydrothermal conditions break down very quickly. If they absorb to mineral surfaces, they last indefinitely. They're very stable on the surface, whereas in solution they're not. We find that minerals play all these roles and more. So we think that it's just one more set of chemical tricks that you can play on early Earth. The idea of that Miller-Urey had of the prebiotic soup, this dilute soup, really doesn't work too well because so many of the molecules don't self-organize and it's too dilute. So you need to have a concentration mechanism. Of course, there are other ways to concentrate too. You can evaporate, wet, dry conditions. You can have an oil slick at the surface. You can have aerosol particles with waves crashing against cliffs and fine particles and go up in the atmosphere where they're exposed to ultraviolet radiation, and they too can hold small amounts of organic molecules. So there are many, many ideas floating out there, but they all involve some kind of selection and concentration and reactive surfaces. It seems like those are essential parts of the story.
Mm-hmm. And actually, the second. Oh, sorry. The second question about is about the first part of the talk. So, the condition that should be met for relevant evolution to take place. So, of course, at some point we need some self-replicating entities, which display some variation. But that maybe what we feel about the idea that we need also the fact that those entities should increase their complexity, which is an additional condition. So we can have selection, which is not relevant selection, which is not going in the relevant direction because, for instance, there's no selection for complexity. They become simpler and simpler.
Yeah.
So what do you think about it? Is there an additional condition about what are the relevant entities?
Yes.
So
Yes, and that's a very, very important point. That's another whole talk that I give about increase of complexity in systems. And I've studied something I call mineral evolution, because this is a classic case of a system that starts very simply and complexifies. So all of these evolving systems, be it nucleosynthesis, be it minerals, be it organic molecules, be it life, be it computers or bicycles, every system that evolves, you have to have a series of rules. First, you have to have a system in which there's combinatorial richness. You have to have a mechanism for sampling that combinatorial richness. And you have to have a selection mechanism which selects for function of some sort, whatever function means. In minerals, it's just simply thermodynamic stability. In life, it could be survival in some kind of environment, which is another kind of thermodynamic stability, but you thought of that way. And so in every one of these cases, the way that you get evolution is by having a system that can rapidly sample the combinatorial richness of its space. The thing that's so beautiful about organic molecules is, as I alluded to, that it takes very, very little for an organic molecule to mutate slightly, just having a different functional group at a different point, having a different conformation, the way molecules can have isomerization. And so you have all these selection potentials within organic molecules, and you have functions which have to do with stability, or in the case of a self-replicating system, the idea that it's more efficient at making copies of itself and using food stocks around it, and therefore suppressing other systems. So I think that these are extremely important considerations, and that once you have a self-replicating cycle of molecules, as long as it can mutate relatively easily, and as long as there's some kind of selection pressure on it, then evolution goes very rapidly, and complexification is merely it's just a factor that goes along with every evolving system. Every system we've looked at, languages, for example, is another case of evolving systems where you have combinatorial richness, you have a selection process, and complexification just follows from the fact that you're filling more and more niches.
Mm-hmm. Because my question was motivated by the fact that as far as I can tell, one typical objection to the self-replicating molecule template first idea is that even if you can have a scenario in which there is evolution by selection, the elements do not become more complex. Not
yes.
So
you need to have
from the
mutation.
very beginning some complexity.
Absolutely, you absolutely have to have a system that's sufficiently complex that it can mutate,
So good with that.
and therefore you have the combinatorial richness. So those three things:
Okay.
combinatorial richness, mutability, and selection. Without all three, you can't have an evolving system.
Okay.
I'm interested, you mentioned something about predicting minerals which haven't been discovered yet. How do you mean, like, to predict their structures?
Yes,
or?
we've actually just published a paper predicting 145 missing minerals that incorporate the element carbon. And the way we do that is we look at all the minerals of carbon and say they're, based on the statistics, there are 145 or so missing. And there's a lot of additional nuance to that, but 145 is the minimum number on Earth today that have not yet been discovered. And then we look at the minerals of carbon plus oxygen, carbon plus hydrogen, carbon plus sodium, carbon plus calcium. And we can say for each of those subsets, we can do the same statistic and say, of the 145 missing minerals, most of them are hydrous carbonates. We can say that a certain fraction of them are calcium carbonates, a certain fraction are sodium carbonates. We can predict where do we see the largest diversity of these minerals. We can go to crystal structure databases and say, here are the synthetic phases that look like minerals that haven't been found yet. And so we recently, just last December, we announced something we call the Carbon Mineral Challenge, in which we announced to the mineral collecting and the mineralogist community the challenge to find these new carbon minerals. We predicted what they are, and 5 of them have already been discovered. So this is the first time in the history of mineralogy that I know of that it made it a predictive science of just using these statistic methods. So that paper's out, and we're doing the same thing for the minerals and many other elements now. We're trying to be as comprehensive. It's a matter of looking and sifting through very large databases and applying these statistical methods. But it's very much the same way that you would do this for birds. If you took all the birds on Earth and you know how many have been found and you have the accumulation curve, you can predict ones that are missing. One of the things that's very obvious is most of the birds that are missing are little brown things that look like other birds. They're not going to be bright orange and green birds that are flying out in open fields in near Paris, because those would have been seen already. So there'll be little brown things that are occurring in the jungles of remote places, and you can actually use statistical methods to predict where to go and find them, and people have done that. Make an expedition and find 10 new species of birds because they know where to look and what kind of things to look for. So it's a lot of fun. I'm sure there are also going to be surprises. That's one thing that's clear. You can't predict everything that's missing because there are all of these sort of anomaly, weird minerals that occur just because of very strange combinations of chemical elements. We also predict, by the way, that there are a potential on all Earth-like planets about 20,000 different mineral species, of which we say 6,500 exist on Earth today. But if you replay the tape of Earth, and that's a talk I'm giving tomorrow at the Institut de Physique du Globe, if you replay the tape, there would be 1,000 or more minerals that are unknown on Earth today, but would arise just because of the statistics of the rarity of the geochemical environments that are weird. If you took all the 10 to the 20th Earth-like planets in the cosmos, that you would find that no two planets are mineralogically identical, that every one has a different suite, but collectively there might be about 20,000 minerals. So this is all kind of fun applications of statistical methods that have never been tried before. All the papers are 2015 or 2016 publication dates so far.
I was asking a question.
Okay, so Nicolas.
So, thank you for the talk. It was very interesting, first of all. And I have 3 questions. So, first question, could you please sum up your position concerning the necessity-chance dichotomy? Because you said that you find that in your opinion, this dichotomy is not relevant. But do you also have a sort of positive position in this sense? And would I be wrong if I said that your opinion necessities could be defined as a computational byproduct of the accumulation of a large number of potentially unique events? Second question: what's your opinion about the so-called anthropic principle hypothesis? Third question: with your analogy, when you spoke about mineral ecology, were you thinking about sort of application of the principle of natural selection to mineralogy?
Excellent questions. And let's see if I can recount each of them. So I do think, because I've devoted the last 20 years of my life to studying the origin of life, my philosophical stance has to be that life is a cosmic imperative. If life is a unique event to Earth, then studying it becomes almost impossible scientifically. It's a miracle, of course, that it's precluded from scientific study. So, yes, I think even though maybe some individual steps are extremely unlikely on the timescales of a laboratory experiment, so therefore it may be futile to try to understand the exact sequence of events that led to our life on Earth. I think that in terms of the combinatorial richness of planets in the cosmos, that indeed life is something that emerges over and over and over again, and that we will find that. But that's just a philosophical stance. It's not at all something I can prove or demonstrate. So you're absolutely right that that's an assumption. The anthropic principle is something I try to avoid at all costs, because I find the arguments ultimately become circular. That obviously, we see the universe from our own perspective, and that the only way that we can be here talking about the origin of life is if life originated and we evolved with it. So I'm not sure what more one can say, other than I think life is a cosmic imperative. And if life's a cosmic imperative, it's because I think that the evolution drive, this idea that we have combinatorial richness, we have selection, and therefore we have evolution. I think the idea of intelligent life arising more than one time is very, very high. Even if we haven't seen it, even if the Drake equation is working against us, I think that's very high. The third question had to do with mineral ecology, and reminded me of the specific nuance you were asking about mineral ecology.
Well, would you when I was interested in knowing whether you think that the principle of natural selection, as it was formulated by Darwin in order to explain some features of the biotic world, could be applied to the
I should show you this. I'm going to just, if you just bear with me for 1 second, I will show you a slide from the talk I'm giving tomorrow, because I think you might enjoy it. I'm talking in that specifically about mineral evolution, and if you will bear with me, this will not take long. Yes, this is a very important point. So, tomorrow's lecture has to do with mineral ecology. And what I want to do in that is look at this question of the diversity, disparity, distribution of minerals. But to do that, I need to start by talking about mineral evolution. And so I have this large number of collaborators. And mineral evolution is something that we started in 2008. This was an idea that was introduced in the paper 2008 that has subsequently become just widely cited and discussed. It's turned out this has really triggered a lot of interest. So mineral evolution means you look at the diversity of mineral species, these 5,000 plus species on Earth, the relative abundance of those in the near surface environment, compositional ranges, also grain sizes and shapes, which really have not been discussed in a long time, although when you go to a museum, you see all those beautiful crystal forms. Well, it turns out each of the different crystal forms comes from a different time period of Earth history, and that hasn't been analyzed. These are interesting ideas. We look at this near-surface environment, the upper 3 kilometers, because that's where we can study it most easily. It's what we can see in other worlds, and it's what interacts with biology. And the punchline of mineral evolution is that more than 2/3 of all the minerals on Earth are a consequence of biology, which is shocking to the mineralogical community. Mineralogists used to think there's minerals and there's life, and they're separate. turns out no, on planet Earth, most minerals arise only because of biological changes to the Earth's surface environment. But here's this comment, and this gets directly to your point. So people have objected, especially biologists, to calling this mineral evolution. And I think it touches on the point that you make. So evolution has several meanings. Change over time has been used widely in geology. We talk about the evolution of meteorites. We talk about the evolution of igneous rocks. There's the implication of complexification, which of course happens in mineral evolution. Early in the universe, there were only 12 different mineral species. We now have over 5,000 on Earth. There's complexification as you go through various processes. Congruency means that each step follows logically from the previous one. And every one of these systems, as I said, involves combinatorial richness, a sampling of that richness through selection mechanism. But mineral evolution is not Darwinian evolution. This is a cartoon that came out in The New Scientist right after the thing, but it's not that. Okay, so it's a different selection mechanism. And this is what I think has confused people. People, again, remember I said in Origin of Life, there's some people who come to it from as mineralogists or molecular biologists or cosmochemists, and each comes with a very different set of prejudices. Well, the word evolution means something very specific to biologists, but I think it's too specific because biological evolution by natural selection is just one example of a system which has combinatorial richness, has mutability, and has selection. And Darwin has described to us in great detail, and subsequent people have analyzed in great detail, how that works. Mineral evolution works in a different way, and it has to do with the fact that an Earth-like planet has fluid rock interactions that are constantly shuffling and rearranging chemical elements. And then as soon as you bring biology into the picture, you change atmospheric chemistry. And as you shuffle and rearrange the elements, those elements are now exposed to oxygen. They're exposed to hydrogen and water in a different way. They have gradients of pH and of chemistry, of temperature that didn't exist otherwise. And so you keep sampling new phase space. You sample new niches, and minerals form as more and more niches arise. But you could use the same kinds of rhetoric for the evolution of languages. You can talk about the same kind of rhetoric for the evolution of Broadway musicals or your favorite pop music form, whatever you want. These are all evolving systems that involve combinatorial richness, mutability, where you're sampling lots of different possibilities, and then there's a selection process of some sort. So yeah, minerals don't evolve by Darwinian selection, but minerals do evolve through other mechanisms that are characteristic of the richness of a planetary system like Earth. That makes sense?
Yeah. So you mentioned that there are some minerals that are only around because of biological influences. Would this mean that from a exobiology, astrobiology point of view, if we could detect these minerals on exoplanets or asteroids, that would be proof that life is there?
Yes. Excellent. Yeah, it took us a while to figure that out. So you figured it out right away. That's great. Not only that, it turns out we believe that the LNRE distribution is a biosignature. That on the moon, we have statistics now for distribution of minerals on the moon, and we're getting them for Mars. And they don't show an LNRE distribution. They show much more parsimonious distribution of minerals, and they're not a few very, very rare ones. Turns out those extremely rare minerals almost always form the kind of localized niches that arise through biological activities. And so the fact that we see an LNRE distribution, we think can be taken as a clear sign of life, a mineralogical sign of life. Now that's something we're still exploring, but it's a really exciting possibility that you can actually use a statistical distribution of chemical phases that are, by definition, inorganic, and use that as something, because minerals are quite robust. They survive a long time, even if life disappears, even if there's so then of course you can say, well, how are we going to do remote sensing to find those mineralogical niches? And I at this point that's way off in the future, but perhaps ultimately we will find that this is an intriguing way of thinking about it.
Minerals tend to be more robust than fossils?
They tend to be more robust than the kinds of fossils that microbes would produce.
Yes.
So if we have multicellular life, then of course fossils become a wonderful guide. Especially if the multicellular life has hard parts, and they've actually built minerals, but then those are minerals. You know? I mean, it's the shells, it's the bones, it's the teeth that lead to the most robust fossils.
It would be more likely if we could analyze minerals from asteroids. Or from
That's something we're really looking into. Yeah. So the only asteroid right now that we can analyze mineralogically closely is the asteroid Vesta, because we have a whole series of meteorites called the HED meteorites that are all presumed to come from that one body, and they have varied lithologies, and so we can look closely at those. And again, in that case, it's great mineralogical parsimony. There's only 20 or 25 different minerals that comprise virtually the entire body, so we don't see the diversity, but we'd have to do much more extensive studies of the body itself in order to find that out.
In mineral evolution, how would you employ selection? Complex, fine, and
Selection is thermodynamic. Selection, the reason you get one mineral phase and not another is because you form a local environment where the temperature, pressure, and composition leads to a new set of stable phases. So it's the diversity of temperature, pressure, composition, space. And composition space, in terms of minerals, there are 72 different chemical elements that form minerals. So you have to think of this as a 74-dimensional problem. I wish I could show you more of this because there's some really interesting facets of this. Let me fast forward and just, so there's 72 different elements, and if you say what minerals form, it turns out, let's see, can I move this quickly through here? Yes, this will work. Getting there, almost there. Yeah, so think about it this way. And I'll just, yeah. So if one plots, each of these points is a different chemical element. The horizontal scale is the crustal abundance in parts per million on a log scale. The vertical scale is the number of species known that incorporate that element on a log scale. It's a log-log plot. What you find is a very rough sort of trend. Extremely rare elements form fewer minerals than extremely common ones. What you're doing here is you're sampling 74-dimension composition, temperature, pressure, space. Think of it as a volume. If you have certain planets and moons that don't have life, that don't have a lot of water, that don't have a big temperature gradient, you have a very small volume you're sampling. As you get Earth with more and more biology, more biological influence, the volume in this 74-dimensional space increases. So there's a lot of details here, but let me show you a couple of interesting trends that we found. And I'm going to skip that here. Here's the moon. Okay? The moon shows the same kind of trend. But here we only have about 20 or 25 mineral-forming elements that are known so far from the moon. The neat thing is the slope is identical. So here's the slope for Earth with a much more diverse mineralogical characteristic. Here's the slope for the moon, where only 55 different mineral species have been identified. So it looks like there's something, the moon is sampling a smaller phase space, but what it implies to me is that the average volume for each mineral species in this 74-dimensional space is more or less the same. Some have larger volumes, some have smaller volumes, but the average is about the same. What we're seeing here, the slope of 0.22, is a fundamental aspect of phase space in terms of chemical richness and diversity. And so there's something else here. Anybody recognize this plot? Island biogeography. This is something introduced by E.O. Wilson and the mathematician MacArthur 50 years ago. If you look at the log of the area of an island and plot the diversity of species on that island, you get a linear plot. Now think about that. In the mineral case, the horizontal aspect was essentially the compositional volume. Here we're talking about an area of an island. I think these two types of plots are exactly analogous. So the biological ecosystem, the species diversity, depends on how much area you have to play with. In terms of mineral ecology, the species richness depends on the volume in the 72-dimensional space you have to play with. It's the same thing, the slopes of this kind of plot. So I think we have I think there's an analogy between the mineral ecology and the biological ecology. That's the whole point of this exercise. And it really comes back to this question about whether you can talk about evolution in terms of what kind of selection rule you have. It really has to do, in this with minerals, it's a selection based on phase space. So I'm working with some theoretical chemists to find out if that slope can be derived from first principles. And if that is true, that's a really fundamentally exciting idea. Then we add pressure to the question and say, when you go to very high pressure regimes, it seems like there are far fewer phases. Well, is that something that we could also model into this? And these become kinds of fundamental questions about why planets behave the way they behave, and things that are going to be similar and similarities for all planets, and also some fundamental differences between different worlds. So we're going in lots of different directions, some of which are not so much biological, but have this characteristic. We're having fun, I'll put it that way.
I have a question by Sebastian.
Sure.
Okay, so he says that assuming that the origin of life requires mineral surface, or this facilitates the concentration of molecules, and knowing the most probable distribution of minerals in the solar system, was it expected that life be carbon-based rather than other elements than carbon, say, for example, silicium?
Yeah.
Just to cite the most common alternative.
Yeah, so I defer to my chemist friends on this. There's a very large literature on whether or not life could be based on any other chemical elements. But carbon is the only element that has this incredible plasticity of form. The idea you could start with a carbon atom and add 4 different bonds. They could be hydrogens, they could be carbons, they could be nitrogens, they could be oxygen. You can keep building up larger and larger structures. It's certainly true that silicon, which lies right below carbon in the periodic table, also bonds to 4 things. But silicon, it turns out, especially in a near-Earth environment, invariably bonds to 4 oxygens in a very rigid way. And then the configurations that these silicon 4-oxygen clusters can form, energetically, there are very sharp valleys. Quartz, SiO2, the beach sand. Feldspar, which combines with an alkali cation and an aluminum. Pyroxene, amphibole, garnet. There are certain configurations that are very, very stable, which means that silicon does not have the plasticity. It doesn't give you many, many structures that are almost the same energy level. Whereas carbon, the difference in energy between different hydrocarbons and branched and things that have double bonds and things that bring in nitrogen and sulfur and phosphorus, the kinds of variation that you want when you're starting to look for the complexity of organic molecules in the biological system. Those energy levels are so different that it's very easy to mutate. Remember what we said about an evolving system. You have to have combinatorial richness. Carbon gives you that. You have to have mutability, and the fact that the energy levels are so close gives you that as well, especially at the temperatures of Earth's surface. And this is something that's critical. If the temperatures are much colder, like the surface of Titan, I don't think you're going to have the same kind of mutability. It's the fact that we're not too high a temperature so these molecules are unstable. We're not too low a temperature so that you don't get the thermal disequilibrium that allows you to form lots of different things. And so there's something about a sweet spot of carbon chemistry at Earth's surface conditions. And I think that's a very important thing to remember. Sure, you can form the small molecules like amino acid in deep space, and you can form them in hydrothermal vents, and you can form them in the mantle. But the big complex things, the goo, that's so important in the origin of life, seems to really thrive at these more modest temperatures of a near-surface environment. And I think that carbon is the only element that gives you that chemical richness, which then can lead to selection, mutation, variation, and evolution. Good. Tell him I said hi, and I hope he's feeling okay.
I think it's,
Hi.
you can hear you.
Hi. Hope you're feeling.
One more question? No? Okay, so I think we can thank you very much.
Thank you
for
very
this
much.
great talk.